Okay, so Bailey, rather than trying to type out an email, what I thought I would do is just do a video and explain this stuff to you because it's easier to explain it in while speaking instead of typing. So first of all, so your answer to the electrophoresis gel question. So summarizing that process, basically what we do first is, and this part doesn't need to be in your answer, this is just for your information. The first thing that we do is cast a gel, which means that we make like a little science jello. And in that science jello, we have this little mold that we pour it into while it's like liquid, just like you would with making jello. And then we put a little called a comb in there, and it has these little there's these little teeth that will sit inside the gel. And then when we're done pulling them out, or like when the gel's set up and hard, we pull the comb out and it leaves these wells. And then we take our gel and we put it in that gel rig that I showed you guys in class. And we fill it up with buffer. And so that will help, that's kind of like some salt water stuff, some special salts. And what it does is it helps the DNA migrate through the science jello that we made in order for it to separate the different sizes of DNA. So what we do is we take our micropipette, we put it in our wells, we plug in the machine. Oh, I'm blurry. And then we set the machine to the settings that we want, and we hit run. And then we wait however long it takes. The one that we did in class, it was like an hour and a half for it to be perfect. The ones that I did over at NAU when I was researching, we'd run them for 10 minutes because we were looking for different stuff. Doesn't matter. But what's going to happen is the bigger pieces of DNA aren't going to travel as far through the gel. The smaller pieces are going to travel farther. And so basically, to summarize the process, we have a gel. We load our gel with our DNA samples. We run an electrical current through. The DNA is attracted to the positive anode. Because DNA is negatively charged. And then we can use UV light to see where the DNA is located on our gel. So make a gel, load the gel with our DNA samples, use the gel rig, which is the little contraption I showed you in class. Use the electrical current to separate the DNA by size, and then view it by using a UV light. Okay, so that is the gel electrophoresis summary. Oops. Okay. And then your other question was, oh no, I forgot. Let me look it up on my phone. Oh, okay, yeah. So about the bacterial cloning. So, okay. So remember that plasmids are small circular pieces of DNA that kind of hang out in the cytoplasm of prokaryotic cells. And what that does, so we can use these to clone our genes. So we have our gene of interest. Let's say in class we used the gene of interest. We use the gene for insulin. okay, that codes for that human hormone insulin. That's and people that have diabetes either make too much or not enough of insulin. And so for those folks that are making not enough insulin, we can just put it in there. But the way that we used to do it was you used to get it from cows. And so because bovine insulin and human insulin are essentially the same. So, but growing a cow is expensive and it takes a lot of resources. So if we can put this gene into a bacterium, it's a lot easier to grow a bacterium, a lot cheaper, a lot fewer resources. Therefore, it's more cost effective. So what we're going to do is use a restriction enzyme to take our plasmid, and we're going to cut it at that restriction enzyme site. And so then that's going to give us, so these are actually double-stranded piece of DNA. So that's going to give us a linear, we're just taking that circle and cutting it so that it lays out straight. I'm not going to cut my hair back. Okay, we also take our gene of interest, put those restriction enzymes, they find that particular sequence of DNA, and now we can glue that gene of interest in here. This end is going to be complementary to here. So we're going to end up with a piece of DNA. It's going to look kind of like, it's going to look like that. Okay. So the three things that all plasmids have to have, plasmids need three things, right? They need a site of origin. They need that because we want DNA to be transcribed and translated. So we need that spot that says start here, start DNA replication here, start DNA transcription here. Okay, so that's our site of origin. That's why we have to have that. The other thing that we need is a restriction site. We need that DNA sequence for these restriction enzymes to say, oh yeah, we got to cut here, so that we can take this circular piece of DNA and lay it out so that we can then glue on our gene of interest. And so that is necessary so that we can have, so that our restriction enzymes can cut, with the ultimate goal of throwing in this gene of interest. Okay, and then the last thing that we need is an antibiotic. Resistant gene. Okay, and then this one kind of doesn't make, like these make a lot of sense, right? But this one, this antibiotic resistant gene, is a little bit, got to think about that a little bit more. And the reason that we need this is that when we take this recombinant DNA that we made, this chimeral molecule, and we go try to shove it in a bacterium, that living creature, that bacteria, is going to be like, dude, I don't want this. What are you doing? But unless it has a gene in here that is useful for it, and the way that we make that useful for it is by giving it an antibacterial resistant gene. And then we grow those bacteria on Petri plates that have that antibiotic in there. And so what it does is it kills the bacteria that didn't accept this plasmid. They're like, we don't want it. It kills them off, but it keeps but then these guys are alive. So we're actually intentionally selecting for bacteria or antibiotic resistant bacteria. And so we're doing that intentionally so that this other gene or gene of interest can be expressed rather than growing a whole bunch of E. coli that are just regular old E. coli. We want the transformed E. coli. So hopefully that makes sense. Thanks for letting me know that you had questions. Feel free to reach out to me with any more questions that you have. I am going to be a little bit busy this weekend, but I will try to respond to you whenever I can. All right. Talk to you later.